Hi everyone, today we're going to look into the link reaction, which is the second reaction of the aerobic respiration process and also the step that precedes the Krebs cycle. So let's look into it. Now in the link reaction, in the presence of oxygen, pyruvate, which are essentially just 3C molecules that we've produced from the previous stage, which is glycolysis, is going to move into the mitochondria. Now, one thing to note is that glycolysis occurs in the cytoplasm of the cell, while for link reaction and the Krebs cycle, which is the step that subsequently acts after the link reaction, occurs both in the mitochondrial matrix. Both of these processes, link reaction and Krebs cycle, occur in the mitochondrial matrix. Now, what specifically happens is the pyruvate molecules are essentially going to be transported by the transport proteins that are located in the inner membranes of the mitochondria and make its way into the mitochondrial matrix. Now, one thing that you should definitely not confuse is confusing this transport protein with an ATP synthase molecule. An ATP synthase molecule, which is involved in the electron transport chain, is a completely different molecule, although they both kind of act as transport molecules. And this transport molecule too, the one that transports pyruvate into the mitochondrial matrix, also uses energy and active transport. They are completely different, so please do note them. Now, what specifically happens in the link reaction is, well, first, as I mentioned, pyruvate is going to make its way into the mitochondria, and pyruvate is simultaneously going to be decarboxylated and oxidized. Now, although this may not really seem to make a lot of sense, decarboxylation and oxidation occurring at the same time simultaneously, you'll see how that makes sense when you look at the chemical equations, but this is essentially going to refer to the removal of CO2 from the pyruvate molecule, and by removing CO2 from a pyruvate molecule, which is a 3C molecule, i.e. a molecule that has 3 carbons, and we're removing CO2, which has 1 of those carbons, we're going to get a 2-carbon molecule. And this 2-carbon molecule is going to be known as an acetyl group. So we're going to get a 2C acetyl group molecule. And also in the process of oxidation, the 2H and 2E electrons that are released in this process are going to be accepted by the NAD plus. Oh, I think the notation's a little bit wrong here. The NAD plus hydrogen carriers, and this will reduce the NAD plus into an NADH and also one other hydrogen ion. A reduced hydrogen carrier is, in consequence, going to be produced by the reduction of pyruvate, which then leads to the release of 2 hydrogen ions and 2 electrons, which are accepted by the hydrogen carrier NAD+, and then creates these reduced hydrogen carriers. Now, after this, the 2C acetyl group, which we have produced earlier on, they're going to be accepted by coenzyme A. Coenzyme A. Coenzyme A is an enzyme, and when we abbreviate it, we're just going to call it CoA. And the goal of this CoA enzyme is essentially to kind of provide the acetyl group a way of kind of getting into where the Krebs cycle is occurring, i.e. it's going to kind of transport the 2C acetyl group to the location where the Krebs cycle is occurring. And when these acetyl group combines with the coenzyme A, it's going to provide acetyl-CoA, which is kind of, I guess, a sort of an intermediary molecule for coenzyme A to transport the 2C acetyl group to the location of the Krebs cycle.